Okay, go ahead.
Bile salts are recycled.
Bile salts are recycled. Yes. Okay. So what we have is we have these bile salts that come from the bile duct from the gallbladder. Okay, and these are helping you emulsify fats, remember? So you're taking huge lipid droplets and you're breaking them down into smaller ones. That's what bile salts are helping you do. And then what happens is, in the distal part of the intestine, in the ileum, you have mechanisms that take up these bile salts once again. Okay? So basically, as this fat gets absorbed into the tissue or into the intestine, now you're kind of left with these smaller balls of fat with the bile salts around them still. But as you reach the ileum, there's a mechanism for reuptake of those bile salts. So the bile salts get taken back up through the intestine, back into the blood, circulates back to the liver, which then goes back to the gallbladder to be recycled again. Okay, so it's essentially all you need to know. And sometimes we end up excreting some, so sometimes it's missed. And so these bile salts can leave in the feces. And that happens. And that's okay, because we have the ability to produce more bile salts. So if the bile levels fall within the body, then we have the ability to produce more and kind of make sure that we always have enough bile in the gallbladder. Now, if you don't eat fat for a really long time, bile levels will decrease. So then if you start eating fat again, if you ate a really, really fatty rich meal, if you're not used to eating it, what can happen is you can get really kind of an upset stomach and get some diarrhea because you don't have enough bile salts to help you emulsify and break down that fat efficiently to be taken back up. But that's the stimulus again then to increase your bile concentration. So bile is a little bit in flux. You don't always have tons of it around, but it kind of matches what your dietary intake is. Yeah. Yes?
So the CCK, does it act on the liver itself when or does the liver realize when there's less bile coming back when it synthesizes more bile?
CCK's main target is the gallbladder. And CCK doesn't really act on the gallbladder itself, but acts on the smooth muscle that surrounds the gallbladder. So a lot of times in those pictures they're not showing you that there's a layer of smooth muscle that surrounds the gallbladder, and that's what the CCK is actually acting on. So it acts on that smooth muscle, initiates muscle contraction, that's what causes it to squeeze and push that bile out. So it's not really acting on the liver, the hepatocytes themselves, it's acting on the gallbladder itself. Yes? Yeah?
That's really a question. So I know it said on the slide sympathetics, inhibit contractions, so they're the parasympathetics, it just wasn't on there, so do they?
Yes.
Okay.
Yes. Yes.
And you were talking earlier about amino acid being activating both glucagon and insulin.
Yes.
Can you explain?
So why does glucagon lead to, or why do amino acids lead to glucagon secretion? Why does increasing in amino acids in the absorptive state also lead to insulin release? Mm-hmm. And the reason is because, okay, so let's say you eat a steak that has lots of amino acids. Okay, amino acids can stimulate insulin release. And insulin causes your cells to take up glucose, so it causes you to have a decrease in blood glucose levels. Now, if glucose levels are high already, because you just ate a bunch of sugar, that's good, because you want to get that sugar out of the bloodstream. But if you only eat steak with no sugar, now your glucose levels are normal, you release insulin, and your normal glucose levels fall to an abnormal state. Does that make sense? You don't want that to happen. So instead what happens is Amino acids also stimulate glucagon a little bit. Glucagon helps you raise glucose levels. So now, if you just eat steak, and now your glucose levels fell a little bit because of insulin to an abnormal level, this will help you restore it back up to a normal level. So it's more of a balancing piece. And this is also kind of argued too. Like some people think that that's why that happens, but that's one explanation for why it happens. But it's typically you don't have that happening. You don't typically have parasympathetics and sympathetics active at the same time. It's kind of an analogy to that. And typically you don't have insulin and glucagon active at the same time. And that's why I say recognize that these are secondary stimuli, like minor stimuli, not the major stimuli. Yes?
Can you go over the storage of absorbed triglycerides versus triglycerides in the liver?
Yes. So this is a super common one that I get all the time, and one that I saw a lot of people had questions on. Okay. So I want you to think about the difference between a chylomicron and a VLDL. So I'm going to ask you guys some questions. This is one that it might help. So sit down and think for a second. Where are chylomicrons made? Does anyone know where they're made in the body? It's okay if you're wrong. Where?
In the intestinal wall tissue.
Yes, okay. So think about where these things are produced. They're produced in the intestinal wall, okay? So here's your intestinal cell. Okay, here's your triglycerides, your fats. Let's just draw some phospholipids and we'll draw a monoglyceride. We'll draw some fatty acids, okay? These are small enough molecules that can diffuse into the cell. This is your intestinal cell. I'm just going to put an eye here. And your intestinal mucosa. So this is your absorptive cell. These things diffuse in, and this cell converts this into a chylomicron. Now, on the outside of the chylomicron, you know you have those apoproteins. Okay? But essentially what this is just a big ball of fat. Right? Now, that big ball of fat does not go into the bloodstream. It goes into the lymph. Okay. So number one, when I tell you, I want you guys to know where are these chylomicrons, where are these VLDLs, these lipoprotein complexes made. I want you to think about where they're made. Okay? So, is there any way that a fat produced from liver cells can make their way into this thing? No. Because it's produced by these mucosal cells. Okay? This thing isn't circulating and picking up other nutrients. This thing is just made in the intestine based on whatever it is you're eating. If you don't eat fat in your diet, you don't make chylomicrons. Okay? So, chylomicrons are made based on the kinds of foods that you eat. So, chylomicrons contain only dietary fats. Whatever it is you eat in your diet, that's what's that has fatty acids or phospholipids, anything fatty, that's going to end up in your chylomicron. If you didn't eat anything like that, then you won't make any chylomicrons. That doesn't mean that your liver won't be making lipoprotein complexes, so we'll talk about that. Now, I was going to send this out in an email too. On your slides, it's either, there's HDLs, there's VLDLs, there's LDLs. All I really care that you know are that the liver makes lipoprotein complexes. That's how I'm going to reference them on the exam. Okay? So, a lipoprotein complex comes from the liver. Okay? So now we have a hepatocyte. Let's draw bigger. And a hepatocyte can make what I'm going to call a lipoprotein complex. This could be a VLDL or an LDL. So just don't get confused. It's not that big of a deal, but just so you know, when I talk about it on the exam, this is the terminology that I'm going to use, and that's what this means. So this thing, the liver, here's a hepatocyte, is making these lipoprotein complexes, and what do they have inside of them? Well, they have tons of fat. The structure of these two things are almost identical. For the sake of this course, you can think of them as identical. They both have lots and lots of fat on the inside. They both have apoproteins on their membranes. They both have phospholipids around the outside. Structurally, they look almost identical. And their function is also almost identical, or is identical. For the sake of this course, it's identical. Sometimes I hate to say that they are the same, but they're not the same, but for this course, they do the same thing. And chylomicrons and lipoprotein complexes, their job is to dump lipids off into the tissues, okay? So both of these things go out of the blood eventually. So the lymph is eventually going to make it up to the blood. So those chylomicrons eventually get into the bloodstream. And what they do is they dock on mostly onto adipocytes. So if you've never seen an adipocyte before, this is what it looks like. Your fat cells are basically almost all fat. So you have a tiny little cytoplasm, here's your nucleus, okay, and this is all fat. So you basically just have an oil droplet inside of these cells. Okay, so this is all triglyceride, basically. Now, these lipoprotein complexes can dock, soak in a chylomicron, and release its contents into these cells, okay? And their contents are the fatty acids and the glycerol backbone. They make triglycerides out of them. Okay, so they dump these things into the fat cell. So, at the end of the day, they're doing the exact same thing. But the point is, where do these fats come from? Well, as a chylomicron circulates, dumps its contents off, dumps its contents off, dumps its contents off, they get smaller and smaller and smaller, right? So now you have this huge ball of fat that's already unloaded all of its fat, and now it's a tiny ball of fat. And we call those a chylomicron remnant. It's just what the book calls them. So eventually these become chylomicron remnants. And the liver says, okay, we have a lot of these tiny little balls of fat. We don't want to waste it. Let's take them back up. I'll use them and I'll repackage it. So this goes back into the liver, chylomicron remnant. So you got lots of them. And it recycles it, okay? And it consolidates all of the fats that are in these little remnants, and it repackages them as a lipoprotein complex. The liver is also doing other things too at the very same time. What other things does the liver convert to fat when you're eating? What do you guys think? Glucose, yes. Okay, so you can convert glucose to fat. Okay, the liver's doing that. So the liver's taking glucose and it's making fat, triglycerides. Okay, that gets packaged into a low density lipoprotein or a lipoprotein complex. Okay, so now we have chylomicron remnants and fat that was once glucose but now is fat in there from our diet. What else can be converted? Amino acids, yes. So if we take in excess amino acids, those can get converted to triglycerides, and those can be stored in the lipoprotein complex. And so now I want you to appreciate that inside of here, even though it's all triglyceride and fatty acids, where they originate come from two different places. Here, can dietary fats end up in here? Yeah, because they come from chylomicrons. Can fat that comes from glucose that's being converted into fat end up in a chylomicron? Remember where this is made. No, there is no arrow from here. This is happening in a dip site back here, okay? These three things can be repackaged into this lipoprotein complex, but the only place that fats come in a chylomicron are from the fats that you take in your diet. Okay, so that's the main difference. What else do I say in that slide? I ask you about where they originate. Let me see. What else does that learning objective say? Does anybody know? I don't have it with me. Or does that answer your question? If that answers your question, that's fine too. So we can move on. Is that it? Also, it's going to be important knowing what those apoproteins do. They help you dock onto the fat cell. They help you dock onto any cell with an aporeceptor. If you don't have an aporeceptor, it's just like anything else. That ball of fat is just going to float right on by, and it won't dock. But if you have an aporeceptor, now you have the ability to hold on to that lipoprotein complex and wait for it to unload its fatty acids. And that's exactly what will happen.
So there was a lab learning objective about the RER, and I think it asked why would the RER increase and why would it go above 1? I'm wondering if there are actually separate answers for that, since I know for the latter one it'd be because of lactic acid, right? But I'm not sure if that also be the answer for the first one.
Well, so that gets to where carbon dioxide comes from. Okay, so what are the two sources of carbon dioxide within our bodies? So one is what you just told me. The later would be from, you said, what was the second part of that? Why would you go above 1? Lactic acid.
Yeah.
Yes. Okay, so that's one source of carbon dioxide because that hydrogen ion is being converted back to, is being converted to CO2. So now you have this CO2 that's accumulating due to lactic acid production. Now, where does CO2 normally come from? Why do we have CO2 accumulating in our bloodstream? Okay, Krebs cycle. Typically when we're exercising, when we're moving, or when anything is metabolically active, what you do is what happens is, and maybe this is good review. Sometimes I don't know if people understand this, but you have your glycolysis. You have your Krebs cycle. Then you have your electron transport chain. So, fats enter the Krebs cycle here. Glucose enters the Krebs cycle after it passes through glycolysis. Okay, fats enter it after they undergo beta oxidation. Okay, now what you get out of this is NADH, and you get FADH2. Now these two things are really important for the electron transport chain. You have to have these two things, otherwise the electron transport chain doesn't work. But as you oxidize glucose, as you break down fat and you oxidize fat, the Krebs cycle produces lots of CO2 in lots of different steps. Okay, so you get tons of CO2 production. That's the CO2 that you're breathing out. So if you can imagine what tissues would be active when you're exercising, things like your muscles. When you're moving your muscles more, what do you need? You need more ATP. Okay, this is where your ATP comes from. If the demand for this goes up, now you're going to have to run this more, right? Does that make sense? You're going to have to run this pathway more. And so what happens is you break down more fat, you break down more sugar, you make more of these two molecules, these reducing equivalents. Now you put them in the electron transport chain, so you have more ATP, but as a byproduct, you produce more CO2, more CO2. So as the tissue becomes more metabolically active, it produces more CO2 because you're basically running these pathways more and more and more and more depending on what your demand is. And so the CO2 comes from your cells. Whatever cells are working out, so Dr. Wu or whoever was biking yesterday, it's coming from his cells. So as you ramp up the workload, you produce more CO2 because you have to work harder. And now there's more of a requirement for a higher amount of ATP. So what causes the respiratory exchange ratio to increase? Initially, it's because you're producing more CO2. Remember, the RER is a ratio of CO2 production over oxygen consumption. So, your for this to increase, this needs to go up, right? This is going up for two reasons. Number one is you exercise more and more and more, your ATP demand goes up, so you produce more CO2 as a byproduct. So this starts going up and up and up. So as you exercise more, your respiratory exchange ratio moves closer to 1. That's because you're producing more CO2, typically through glycolysis when you're burning sugar. Okay? But then you reach a point when you start producing lactic acid, and now that lactic acid is getting converted to CO2 in the blood, and that CO2 is being added, and now you have even more CO2, and so that number goes above 1. So there kind of is two reasons why that would increase. One is because of lactic acid, and one because as you work higher at a higher and higher level, your demand for ATP goes up, and as a result, you have more carbon dioxide production.
So oxygen would be used more too, right?
Absolutely.
Just like sort of
Yes. So this is where oxygen fits in. So oxygen is happening here, being consumed here in the electron transport chain, and so oxygen is required in the electron transport chain to make ATP. So oxygen gets converted to water, and that helps us make ATP. I can draw that out for you, but that's too much biochemistry, I think, for right now. So this is the general idea. Oxygen is consumed to help you make ATP. So eventually it gets converted to water, but the idea is that it helps you keep up with that demand. If you don't have oxygen around, now you don't have the ability to run this system anymore. So what you have to do is you have to run anaerobic respiration, which produces lots of lactic acid. Not very efficient. The only reason we can grow to be the size that we can are because we have those mitochondria. We have the ability to make lots and lots of ATP. Making ATP from lactic acid production through glycolysis is not very efficient. So it's really important that we have this ability to kind of do this. Does that answer your question?
Yeah, but I guess also like if the ratio is increasing, then like CO production is happening more than oxygen usage increasing, right?
So you're saying as this goes up, then this is also going up? So why is the ratio eventually increase?
Yeah.
So because this is being produced there's more carbon dioxide molecules per round of the Krebs cycle than there is oxygen being consumed. Yeah, that makes sense. I didn't really think about that, but that totally makes sense. Mm-hmm. Yes.
And I guess just going off of that, and the first question, so then if you're under 1 but the RER is increasing, that just means you're increasing your glucose expenditure?
Consumption, like you're primarily using glucose as a substrate?
Yeah.
Yes. So where those RER values come from is what I showed you yesterday when we broke down glucose in terms of the amount of CO2 that it takes to break down, or the amount of oxygen that it takes to break it down, and the amount of CO2 that's produced for every molecule of glucose that's broken down. That's where those numbers really come from. And so if they're equal, that tends to be, that's for glucose. If you're primarily oxidizing glucose, you are producing and consuming about equal amounts of oxygen to carbon dioxide. Now, if you're burning primarily fats, so that kind of goes against what I just said to you. If you're burning glucose, carbon dioxide and oxygen production are produced in equal amounts. If you're burning fats, it's different. It takes a different amount of oxygen, a different amount of CO2. It takes more oxygen, actually, to burn it. And so that's why your respiratory exchange ratio is below 1. Does that make sense? Yeah. So if I were to ask you on an exam, I wouldn't ask you about the middle numbers. I would ask you about one side or another. I'd ask you, does that make sense? Because you can't even tell me accurately what you're oxidizing if I gave you a number like 0.8. It could be fats. It could be a mixture of glucose, fats. It could be primarily amino acids. Like, you don't really know. So I wouldn't ask you that because it's not accurate. Yeah. Somebody else up?
I just have one, so with the liver and like the supposed to absorb glucose? Does it burn amino acids until for like its own energy?
Mm-hmm.
Okay.
Yes. Yes. Yes.
So this is just like a side question. For the liver, you said that from the gastrointestinal tract it gets the deoxygenated blood, right? So for what I learned so far is that you must only rely on oxygenated blood to function. Is it
Yes.
still getting oxygenated blood?
Absolutely. So if you remember, so the question was, there's a deoxygenated hepatic portal vein that goes to the liver. And remember, one of the slides I talked about, there are 3 vessels that you need to know about that are going into the liver. And so those 3 vessels, one is the hepatic portal vein. And I showed you why that vein has to go to the liver, because it's 2 capillary beds connected in series. So for whatever reason, the way that we've evolved, we have, this is your GI capillary bed, and this is where gas exchange occurs. So this is your oxygenated blood, this is LASPO2, and now this is your deoxygenated blood. Okay? As it goes through the GI tract. The oxygen gets picked up by the GI tract and it's used. The deoxygenated blood goes to the liver. Now, this is one vessel that goes to the liver. So this is the hepatic portal vein. Okay? That's deoxygenated. It's deoxygenated, but it contains lots of nutrients. So, deoxy, high in nutrients. Now, what they're not showing you in this picture is that the liver also receives an oxygen supply, because you absolutely have to have an oxygen supply to live, and otherwise that tissue would die. So there is also an artery that goes to the liver. So here's your liver. You have the hepatic portal vein. You have a hepatic artery. Okay? And then you also have a bile duct. Okay, so these 3 things are working in the liver. The hepatic artery carries your oxygenated blood. Your hepatic portal vein doesn't have much oxygen in it, but it has lots of nutrients. Now, this all eventually dumps into the central vein here. So, as the hepatic portal vein comes in, it delivers all of its nutrients. Okay, so now this cell gets some nutrients. The cell also gets some oxygen, which makes it happy. And the cell's also producing bile. And so that bile goes into the bile duct. Now maybe this is better shown in the actual image on my slide. Let's plug this in. So the liver is just kind of weird in that way. 3 different vessels. Normally, you just have an arterial system coming in, you exchange gases, and you do whatever, and then you have a vein leaving that goes back to the heart. But in this case, you have a portal system coming in, and you have an arterial system coming in, and what you have leaving is just a vein. One big vein, because the hepatic portal system, the hepatic portal vein, and the artery are going to mix. Their blood is going to mix and then eventually dumps into the portal vein. So let's look at that a little bit closer. Here. Alright, it's going to take like 2 seconds. All right, come on. Okay, so here you are. Here are your three vessels that I want you to know about. You have the artery, that goes to the liver. You have your vein. This just came from the GI. And remember, that vein had nowhere to go except for the liver, so it has to go to the liver. And then lastly, you have your bile duct. So here's your hepatic portal vein. This is carrying very nutrient-rich blood. It's not oxygenated, but it's very nutrient-rich. That's why the liver kind of has first access to any other organ, over any other organ, to both glucose and amino acids. It's because that blood that just left the GI has nowhere to go but the liver. Okay? But there's a separate branch of oxygenated blood that goes to the liver because, yes, the liver does need oxygen. And then we have these bile ducts. So what I wanted you to know are kind of which direction are these things flowing. That's important. So we have oxygenated blood that's flowing, and it's hitting every single cell in this liver lobule. Okay, so every cell in the liver experiences oxygenated blood. At the very same time, every cell is also experiencing deoxygenated blood, but that's nutrient rich. Okay, so this is how the liver gets access to nutrients on the first pass. This is how the liver gets access to oxygen. Okay, so as the oxygenated blood and deoxygenated blood pass through, they actually mix. So there's some mixing event that happens. That's not typical, but that is what happens inside of what we call these sinusoids, which are really, really leaky capillary systems. And so lots of the nutrients get absorbed, and lots of the oxygen gets absorbed into all of these different hepatocytes. Now, hepatocytes are also making bile. That's the other job that they're doing. So they produce bile. And the bile that they produce, they have to put somewhere, and they put it into these cannuliculi. Okay, and so these basically run the length in between these cells. They're not shown all over, but basically every hepatocyte has access to a bile duct. So it makes bile, dumps it in the bile duct, and the bile runs in the opposite direction towards the bile duct. That bile duct eventually makes its way to the gallbladder, and that's where you store the bile. Okay, but look here, the hepatic portal vein and the artery, they mix their blood, and eventually goes into the vein. And where did the vein go? It goes back to the heart, and the heart pumps that blood around. Okay, that's why there's three different vessels. It's not as common. That doesn't happen very often in many places within the body. But this is kind of a unique structure, and that's kind of why we bring it up and talk about it. Yes.
At one point you say conjugated toxins are also stored in the gallbladder.
Mm-hmm.
So is this where that exchange?
Exactly. So conjugated toxins also are produced or are made inside of these little liver cells. And so those conjugated toxins absolutely get dumped into the bile duct. That bile then moves to the bile duct and moves to the gallbladder. That's how it ends up in the gallbladder. And then that gallbladder, when it squeezes, goes right into the intestine, and you excrete that out. Exactly. Thanks for bringing that up. Some conjugated toxins, though, too, remember, depending on their size, there was a size exclusion that we talked about, depending on their size will end up in the blood and be excreted by the kidney. So sometimes it'll go into these bile ducts, but sometimes it'll get pushed into the blood and go back to the heart, and then the heart will push it to the kidneys, and then those toxins can be excreted by the kidneys. So there's kind of two fates for those, depending on how big the toxin actually is. Yes?
Can you go over the negative feedback system of insulin?
Yes. Okay. So, let's... Okay, here's kind of a busy slide, but I feel like it tells us everything we need to know. I'll walk you through it. All right, so you eat a meal. Insulin, again, you should know that that's dominant in the absorptive state, right? Insulin really is only around when you have nutrients around, when you have high levels of sugar. So you eat a meal, eventually that sugar gets absorbed, and so there should be an increase in sugar. So nutrient digestion absorption, you have an increase in plasma glucose level. That is the primary release or stimuli for insulin release. So that goes to the beta cell and says, release some insulin, we have high levels of glucose. So think about, that is the primary stimuli. So when you think about negative feedback, what you're trying to do is change that primary stimuli. You're trying to bring that glucose level back down to normal. That glucose level is out of the normal range. It's above that 5 millimolar range, or that 1 teaspoon of sugar. Okay? So when that plasma glucose level gets too high, you activate the insulin release. What is insulin's main job? Insulin's main job is to activate lots of different pathways that decrease plasma glucose levels. And so when your glucose levels fall back down, that's your negative feedback that then resets the system. Okay? Does that kind of make sense how insulin activates pathways that feed back negatively to decrease glucose levels? And now you have now your glucose levels fall back to normal. And if your glucose levels are normal, this stimuli goes away, so you don't stimulate the beta cells, insulin levels go down, and everything's happy. So it waits for the kind of the next stimuli to activate the next pathway. I can see what other questions you guys had.
Can you do Hirschsprung's disease?
Yeah, Hirschsprung's disease. Yes. Okay, so the basic idea here is that okay. So normally for you to not only move feces around, but for you to defecate, you have to have a nervous system intact. Okay, you have to have nerves that do a couple of different things. So the nerves are going to help you push the food along the digestive tract. And it's also going to help you relax the internal anal sphincter, which is really important for you to actually defecate. So if you don't relax that internal anal sphincter, you have a really hard time defecating. And that's kind of your stimuli to know that you have to go to the bathroom too. Because now you have feces pushing on your external anal sphincter, and that's usually what tells you, okay, I need to go to the bathroom now, and you decide when it is that you need to poop. Okay? So what happens in Hirschsprung's disease, Hirschsprung's disease, which is kind of interesting, is that they're missing a collection of nerves, typically in the distal part of the colon. Now, if these nerves that help you relax the internal anal sphincter are not there, you don't have the ability to relax that sphincter and go to the bathroom. Okay? You also have problems with motility. So you have problems moving that feces along. And so what happens is it kind of gets stuck at the distal part. So right before wherever those nerves get cut off, this feces doesn't have the ability to move as well anymore. Okay, so it kind of fills up, and so what you get is this enlargement of the colon. That's called megacolon. And now, in order to move that feces down, a lot of times what they have to do is they have to be on lots of laxatives. Okay, softens the feces so that it's easier to pass, easier to move. People tend to have to really bear down when they go to the bathroom, which is really uncomfortable, but you're trying to force open that internal anal sphincter that really doesn't want to be forced open. So it can be really, really uncomfortable, and most people suffer from severe constipation. So the excitatory piece of the ENS would be the pieces that are helping you with motility. The inhibitory piece is what happens is that that sphincter is tonically contracted. It's tonically in a contraction state. And so what you need to do is you need to activate an inhibitory neuron, and that inhibitory neuron relaxes the muscle. So it does the opposite. It inhibits the contraction, it relaxes it, and when you have this disease, the excitatory and inhibitory neurons are missing. So not only do you have problems with motility, but you also have problems with relaxing the internal anal sphincter, and now you can't defecate. Yes?
Could you go over the differences in the absorption of carbohydrates, proteins, and fats?
Yes, so that's a little bigger one. Okay.
same thing as like determining the fates of all those things? Is that kind of saying the same thing? Or is that-
This is absorption from the inside of the intestine to the blood. Is that what you're asking about? So movement from the intestine into the blood is different from the blood to the tissue, where it's going to be long term. So yeah, there are different pathways, and you don't have hormones around. These are just basically based on transporters that are present on the membrane. So I can kind of tell you basically what I want you to know. So you have carbohydrates. The food that you eat, typically when you eat carbs, is in these long chain-like structures called glycogen. And we have amylase that's present. Amylase is present in a couple different places in our body, in our mouth initially. So you start starch digestion in the mouth. You also continue starch digestion in the intestine. So you don't have amylase in the stomach because it becomes inactivated. So amylase helps you break down this glycogen into these kind of disaccharides. Disaccharides means 2 glucose units kind of put together. But there's different flavors of glucose units that can be put together. So you can have fructose, you can have lactose, and you can have glucose. And depending on the combination, you get these different sugars. So lactose is 2 glucose, or glucose and lactose. Maltose is a glucose, or 2 glucose molecules. And sucrose, your table sugar, is a fructose and a glucose molecule put together. Okay, so all I really want you to understand is that we have enzymes for this. We have enzymes that help us digest that starch down. I don't care if you need to know. If you know all the enzymes, that's not important to me. Amylase is important because that's something that we talk about in multiple places within the gut. We talk about it in 2 different places, and it comes up again here. And all I really care that you know is that it helps with starch digestion. Okay, it's an enzyme that helps with starch digestion. And I pointed this out a couple of different times in class, but the reason why in your mouth amylase doesn't help you break the glucose all the way down is because it can't. See how it's breaking glucose down to these 3 disaccharides? It doesn't have the ability to break things down to the monosaccharide level, so we need additional enzymes present, and those come into play when we get into the intestine. So you can have the initial breakdown in the mouth that starts digestion, but it's continued in the small intestine. Okay? All right. Now, here's this gets to be where I can start asking a lot of different kinds of questions. Okay. So there's a couple pieces that I really care that you know about here. Number 1 is if you see glucose riding along with sodium, what kind of transporter is that? Secondary active transport. Okay, that's important for you guys to recognize. So it's important because it's telling you that glucose is in low concentration here compared to here. I hope that you can see why that is. Because you're using the energy as sodium moves down a concentration gradient, releases some energy. This little transporter harvests that energy and helps you move glucose up a concentration gradient. Okay, that's secondary active transport by definition. So, if I ask you a question on the exam about glucose levels on this side versus this side, I would hope that you would understand that they're higher on this side than they are on this side because you're required to use this transporter. Now, if you think about where you are in the intestine, if you're in the beginning parts of the intestine, you just ate a milkshake, let's say, so you have tons of glucose around. And glucose levels outside are higher than on the inside, do you need that transporter? No, you probably don't. So, there are also other kinds of transporters that are present. You have a facilitated diffusion transporter that could be present, and now glucose should just move in through that transporter. Alright, so the one thing I want you guys to think about is, what are the concentration gradients? Because when you look at those channels, I want you to automatically thinking about what the concentration gradients are. And 2, sodium is very, very commonly used to bring substrates into the cell. Okay, substances like glucose, it's used to bring hydrogen ions, it's used to bring all different sorts of things into the cell, amino acids. And so what this is showing you is that glucose moves from low concentration out here, we don't want to waste any, into the cell where it's higher. And then it's moving through facilitated diffusion on the basolateral side, which suggests that now it doesn't need energy. There's no input. So it's moving from high to low. So it's moving from low to high, and then from high to low. And so I want you to be able to tell me then which side has the lowest and which side has the highest amount of glucose. Those are kind of that's physiology that I want you guys to think about beyond what's going on in the actual picture. And those kinds of things are going to come back with cell That kind of information is important here. It's important when you're moving substances into the liver, like glucose. Things are moving at a concentration gradient, so when you see these types of transporters, I always want you to think about, why is it secondary active transport? Why is there facilitated diffusion? Could it be facilitated diffusion? When would that kind of happen in the body? This is showing you one snapshot, and that's not always extremely accurate. So I think just thinking about those kinds of things is important. So that's what I would focus on. That sodium comes in down a concentration gradient, and you know there's a sodium-potassium ATPase that kicks it back out. And so why does it do that? It's so that sodium levels remain low, so that sodium can continue to come in and bring other things in. If this thing died, now sodium wouldn't be able to bring in glucose anymore, and you'd end up wasting lots of glucose in the feces. Okay? Fructose I don't care as much about. It also comes in through a facilitated diffusion transporter on either side. Again, the thing that I would care more about in this case would be thinking about which side would have to have the highest amount of fructose in this case. So fructose appears to be highest here because it's moving down in concentration, and again down in concentration. So highest on the outside, medium fructose levels, lowest. Those are the kinds of things I think are important. Glucose, on the other hand, is a little bit more important because it's a molecule that we talk about over and over and over again. Okay? Okay. All right, protein. On the exam, I will always say brush border enzyme, so that you know what that is. A brush border enzyme is an enzyme that breaks down zymogens. So basically, a zymogen is a protein, an enzyme, that has additional amino acids attached to it. It's very analogous in a lot of ways to steroid hormones that ride on the back of proteins. The proteins can kind of shield them, protect them from being chewed up by other enzymes. When you have zymogens, it's basically these extra amino acids that protect them from being active, overactive. And so what happens is you have to remove those amino acids before you can activate the enzyme, and that's what those brush border enzymes are doing. They're basically cleaving off these extra peptides and activating the enzyme. And so also you don't want a bunch of active enzymes coming in, like leaving and being active in the pancreas, and the pancreas has lots of different ways to protect itself from enzymes that accidentally get activated where they're not supposed to be. And so there are different mechanisms that actually target trypsin in the pancreas if it gets activated, and it kills it right away so that it can't cleave out the pancreas. So you want trypsinogen to come from the pancreas, make its way into the intestine before it gets active. And the thing that activates it are these brush border enzymes. Okay? Different books call them different things. I'm going to call it a brush border enzyme. Trypsinogen then gets cleaved, and you form trypsin. Now, once trypsin is active, trypsin is the most important, probably, enzyme because it activates all the other ones. If trypsin goes away, now you have a harder time activating the remaining zymogens, and that becomes a problem for you if you need to digest the food that you're eating. And so trypsin activates all the other enzymes, and now you have all of these enzymes that are active, things like pepsin, trypsin, chymotrypsin, elastase, aminopeptidase, carboxypeptidase, all of these become active, and what do these things do? I don't care that you know the names of these. I care more that you understand that amino acid, or proteins are these long chains of amino acids, and they need to get broken down. Okay? We have smaller peptides, and we also have individual amino acids, and in between here we also have di and tripeptides. But I'm not showing you that. And those get absorbed differently. So again, on an exam, what I really care about, brush border enzymes that activate trypsin, know what a zymogen is, know what activates zymogens, this is also a zymogen, know what activates trypsinogen, and then what activated enzymes do. You don't have to know what all of those enzymes do individually, but in general, what are enzymes doing? And what are they helping you do? And so for proteins, they're helping you break down long proteins, or large proteins, into peptides, and eventually into di and tripeptides, amino acids, and small peptides. Now, this is also showing that the vast majority of the absorption is happening through amino acids. So most of these peptides are being broken down all the way down to amino acid levels. Some of them are being broken down to dimetripeptides that can be absorbed, and a small amount, because this is very energy taxing to move large peptides across the membrane or across the cell. Typically don't do that very often. So only a small amount is absorbed that way. So from this slide, what I really care that you know is that you can absorb amino acids in or protein in 3 different ways. I don't really care as much that you know, I would say amino acids because they're the most important is what I would focus on. So understanding that amino acids are moving into the cell and how they're moving. Again, moving with sodium, so understanding what kind of transporter that is. That's important. Again, if I ask you about concentration gradients, you should be able to tell me which side has the highest concentration gradient or has the highest concentration of either the amino acid, the dimetripeptide, whatever it may be. Those are the kinds of things that I like to think about. But knowing that these 3 methods do exist is important. Like I could easily ask you a question about that too. Oh, and then we have fat. So bile salts, again, break up the fat, but they don't break up the individual triglyceride. That's not their job. So let's just look at this. Whoa. Okay. So if we just go through, we have the fat droplets, bile salts help to break that up. We have pancreatic lipases. Remember, these are enzymes that are coming from the pancreas that help you break up fat. And what this is doing is it's helping you so that you can release, it's breaking the triglycerides. So here, the lipase are taking those triglycerides and it's breaking it down to your diglyceride, monoglyceride, and your fatty acid. Okay? And that's being stored in these little micelles. So inside of here now you have triglycerides that are kind of torn apart. And then as these approach the membrane, those little triglycerides or those pieces can diffuse out and across and into the cell. And that's kind of what that's showing you. And then once it's in here now, look what we make. We make a chylomicron. All of the fats that are in a chylomicron come from whatever it is that you've eaten, because that comes from the intestine. Those chylomicrons then exit the cell, too big to fit into the capillary, so they don't move into the capillary. They move into the lacteal system, into the lymph system. The lymph system eventually makes its way back up with the blood through the thoracic duct, and then makes its way into the vena cava, back to the heart, and then it gets pumped everywhere in the body. But every time you move fats in and out of a cell, you have to break them apart and then put them back together. It's just kind of energy taxing, but it's the only way to do it. And then water and ions. The main thing I want you guys to recognize here is that as ions are moving across the intestinal cells, you create a difference in concentration gradient for water to move. So the osmolarity here, as sodium moves, as chloride moves, as more sodium moves with glucose, the concentration, the osmolarity on this side goes down, which means the water concentration goes up. And so as sodium leaves, water concentration increases, causes water to move down a concentration gradient. And this is why water tends to follow the movement of ions. So whenever ions leave an area, now water concentration has gone up in that area, so now water leaves and follows. And the same thing. So as water moves across, so now as these ions make their way over here, now this becomes really concentrated, and now water can move down a concentration gradient in. Does that make sense? Mm-hmm. So that's why if you get a cholera toxin, or you get a toxin that targets one of these transporters, so I think cholera toxin targets this transporter right here. And what happens is people get massive diarrhea. And the reason is because now you're not absorbing as much sodium and chloride, and so that sodium and chloride is staying in the intestine, less water is leaving, and that water leaves in your feces, and now you have diarrhea. So how do they treat that? So if there's a problem with this transporter, what they do is they typically give sugar and salt in water. So why would they do that? They give sugar and salt to activate this transporter. Overactivate this transporter to kind of make up for the lack of this transporter. When you do that, now more sodium, more glucose gets pulled in, you pull more water with it, and now you can try and rehydrate these people whose channel is kind of blocked or not functioning as well until you can get rid of the cholera toxin. That channel comes back to life. But that's typically the oral rehydration therapy that they use is a glucose sodium load, which is also, I was talking to you about this, but which is why sometimes if you think about what you should drink if you've ever had too much alcohol, the best way to absorb more water is, so having a little bit of salts in your water is a good thing. Salts and sugar is a good thing because then you'll absorb more water across your epithelium instead of just drinking pure water. If you just put tons and tons of water on the side, yes, you might have some absorption, but if you add a little bit of salt, now as you move, you're going to pull more and more water with it. Okay? Which is why Gatorade is so hydrating for people. That's why they have Gatorade on the sidelines all the time, because it has a little bit of sugar, it has a little bit of salts there, it helps you move water in. Okay? Yes?
Are those sugar enzymes considered fresh water?
Sugar enzymes. Amylase? No. So brushwater enzymes are different. They're very specialized enzymes that really are only involved in activating zymogens. Okay, that's the only way that I want you to think about them.
Are they like attached?
Yes, they're attached to those villi that kind of sit inside of the intestinal mucosa. Yes?
So when we talk about like, I was just like, this more to clarity thing, so like, the submucosal plexus, like all those cells, the sexual secretory cells that are on the mucosa. So it's like they're like disconnected by like, so when you're like secreting, it's from the mucosa. I'm just
So they're right next to each other. So yes, the mucosal plexus is going to, so the glands kind of sit within the submucosa. So yes, so when you activate the glands, they kind of move through both like the epithelial layer and the submucosal layer. So yes, so it's innervating within the submucosal plexus. If I ask you about that on the test, I will be specific. If I ask you about submucosal plexus activation, I always want you to think about some sort of secretion that's happening. Okay? Don't choose muscle contraction. It's not the right answer. Okay? So I want you guys, and if I talk about my enteric plexus activation, don't choose a secretion, because those two things are doing they have two different purposes, and for the sake of this class, that's as far as I really want you to know. Yeah. Yes?
There's like one other thing. I was just looking at this slide. For gastrin, so amino acids, that causes gastrin release.
Yes.
And it also causes reduced motility of the stomach.
Why? Where did I say it inhibits motility?
It was just on this slide.
So amino acids will activate the release of CCK. CCK can inhibit motility, but that's from the small intestine. So once those amino acids hit the small intestine, then that would inhibit it through CCK, moving through the bloodstream, acting back on the stomach to inhibit motility. So I don't know if that's maybe what you're thinking.
It says fat acid. It's like fat and acid.
Yes, absolutely. Yeah, so both. But I was just thinking about amino acids when you were asking me about amino acids. But yes, both would activate CCK release. CCK goes to the gallbladder, CCK goes to the pancreas, and CCK goes to the stomach. It has all different effects in all three places. Yeah. Yes.
Can you explain like the release of enteral endocrine cells and what CCK, like how that reflects yours?
Yes. Okay. So let's see if I can go to my slides here. Okay. for CCK?
Yeah, or
Okay. Both are working very similarly. Okay. Where's CCK? All right, let's start with Secretin because I feel like this picture is a little bit better. Okay, but the mechanism's essentially the same. It's just the stimuli is different. So for secretin, you have these little enteroendocrine cells that release secretin. But, so here we are in the intestine. You have high hydrogen ion in the intestine in the initial part because it's just come from the stomach. Here's the stomach. You've entered into the small intestine. You have high hydrogen ion concentration, and so we need to do something about that. We need to neutralize that. And so what happens is these little enteroendocrine cells, they're the ones that have these little sensors. They have chemoreceptors that sense hydrogen ion. They become activated and depolarized. The enteroendocrine cell itself says, okay, there's acid here. I'm going to release the hormone that I have inside of me. It's called secretin. And secretin is a hormone, so you should immediately think it goes into the bloodstream, which it does, which is what I'm showing you at the bottom. And then in the bloodstream, that goes everywhere within the body. So anywhere that has secretin receptors, that will become activated. And here I'm showing you that it goes to the pancreas. And in the pancreas, it signals for bicarbonate release. All right. And now bicarbonate leaves, helps you neutralize, everything's happy. Now the same thing is happening essentially with CCK. So when you have fats and you have amino acids, glucose is thought to do this a little bit too, but you activate these endocrine cells, you release CCK instead of secretin. Stimuli is different. The same exact cells have different receptors for different things. That goes back to the pancreas, activates the pancreas, and now you get enzyme release. So both are kind of doing the exact same thing. Now, that secretin also is in the blood, and when that blood goes to the stomach, there are secretin receptors there, and that can inhibit motility. So that's kind of what we were talking about before, where you can inhibit motility in the stomach because there's secretin or CCK present in the blood. Does that help? Yeah.
So, like, I was wondering how, like, does it use a pancreas system or not?
In this case, no. It's this secretin is being released just so the cell itself is kind of its own integrating center. The cell itself is saying, there's a stimulus, is it high enough for me to release secretin or not? So this is independent of the ENS. That's why this section is really hard to teach, because also 100% guarantee that the ENS can control those cells and secrete secretin too. So yes, I guess the way that I've taught it is that it's independent of the ENS. So yes, I guess that's where I'll leave it. Let's see what else do we have here. It's also important to kind of know where things are acting. So it's important to know that different cells are releasing different things. So the pancreatic islets, so these are the beta cells and the alpha cells that we talked about, and the delta cells with glucagon, with insulin, with somatostatin. Those are hormones that are released, and those are released by the pancreatic islet cells. Any exocrine function, you're going to put something into a duct, that's going to be released from from the acinus, which are either the acinar cells, which are these pink cells up here, or the duct cells, which are these cells down here. Duct cells release something different from the acinar cells. So duct cells secrete bicarbonate. So these guys are responsible for secreting bicarbonate. These guys are responsible for secreting digestive enzymes. So keeping those two things straight is important too. And it's also important because thinking about diseases, we didn't talk about this that much, but depending on, you can have blockages of these different, of the ducts. Depending on where the blockage occurs, you might see there might be different deficits. So if you have blockage up here, you wouldn't be able to produce digestive enzymes, but you'd still be producing bicarb. So you'd still have the ability to neutralize your chyme, but you wouldn't have the ability to break that chyme up as well. Does that make sense? Whereas if you had a blockage down here, Now you're not making any bicarb, or not releasing any bicarb, and you're not releasing any digestive enzymes, so your symptoms in your patients are going to be different depending on where those kind of blockages occur. Okay, so that's why it's kind of important to know where those are coming from.